Okay, good morning to all of you. So today we are observing the Xanthophyta, Chrysophyta and Basilarophyta. So here in Xanthophyta, these are yellow-green algae or these members. They are the important as a heterocont algae. That is, different types are there, as they found in the fresh water, also in marine water and the soil habitats. So therefore, they are to be heterocont are there. The single-celled flagellate to simple colonial and filamentous forms are there. The xanthophyte, they shows the chloroplast and they contain the photosynthetic pigments, chlorophyll A, chlorophyll C, beta carotene and the carotenoid diadinoxanthin. As these are the heteroconts, so their chloroplast do not contain the fucoxanthin and which accounts for their lighter color. Therefore, the storage of polysaccharide as it is in the form of the chrysolaminarin. So remember all these important highlighting characters for the Xanthophyta. Xanthophyte cell walls are produced of cellulose and the hemicellulose. They appear to be the closest relative to the brown algae. Here you are observing the good example of the Botryidium. You remember this name of the genus. It is the Botryidium. Species is the Valrothi and second is the Miscoccus. So these are two genera mainly. So here you see how is the plant body is there of the Botryidium. So it shows the vesicle-like structure, rhizomorph or the rhizoid-like structure. Then their gametes are there, or these are the single-celled flagellated. So it is very interesting form granulatum. And second is the chlorothecium. Chlorothecium is the single cell for this colonial like structure. So from unicellular to colonial structure. So this is the another example. Nextly, this is the Botryidium Valrothi is there. Then micro Miscoccus is the one another genus. So see just how is the nature. Then Chlorothecia is the fifth one. So remember the proper names of this genera. Then here the species now placed in the Xanthophyceae were formerly included in the Chlorophyceae. In 1899, Luther created a group as a heteroconti for the green algae and with equal flagella. Pasteur in 1914 included the heteroconti in the Chrysophyta. So again in 1930, Alorgay renamed the group as the Xanthophyceae. So here these are commonly they are to be having the monodoid, that is unicellular flagellate, and also sometimes the amoeboid species which have been included by some authors in the protozoa or the protista. Again the heterochloridina according to Doflin and Ricon in 1927 to 29 as class Xanthomonadina with order Heterochloridia and the Rhizochloridia. So it is given by the different scientists. So there are the many views are there. So therefore I am explaining these all changes here. According to Holmberg in 1964, the Heteromondalia or literally in 1983, the subclass Heterochloridia. So just we have to know about all these characteristics. Then our main scientist Fritsch in 1935 so recognize the following orders in this class Xanthophyceae. So order Heterochlorideals. Then there are 4 suborders with examples based on the genus name. So Heterochlorosis, Chloroidaceae, and Chlorogloia. It is named as the Heterocapsaceae. Then Miscoccus family Miscoccaceae. Then Heterorhizoidea based on the genus Rhizolecane. Then order Heterococcales. Same, these are the different examples. Haplospira, then Microchlore. So please you remember all this given by the Frisch for the Xanthophyceae. Then Botryddium is in the lastly, the Botryddiaceae. Now we are observing there is having the heteroconty. So this shows the different examples here for the algae. So you just see this is having the Ophicythium, then Botryddium erizi, right? Conifera minor, it is unicellular. Then Conifera bombicina. So like this one, these all genera. Just we have to remember this one. This is the Botryddium arbuscula. So Botryddium species shows the different structural or the habit nature. So they are commonly also found in fresh water and also the marine water. So therefore this is the one part. Then in Chrysophyta, Chrysophyceae is usually called as chrysophytes. So therefore these are golden brown algae or the golden algae. Here as you are observing, mostly they are found in the fresh water, and therefore the golden algae refer as a single species. It is having the Primnaceum parvum, which is causes the fish kills. Then Chrysophyceae should not be confused with the Chrysophyta, so which is most ambiguous taxon. In Chrysophytes, as there is a ugly suggestion of the Chrysophyte is there, that is which refers to the Chrysophyceae. Some pond members are there. Hikorima, algae of the light. It is given by the Japan scientist Hitachi. It is an uncertain genus of the Chromeluna. Then Ochromonas or the Chromophyton. It is a commonly called as a golden algae. So you see just how is the habitat of this Chromulina is there, and it is very interesting. So it is shown here magnified part, and some sketches are there, right? Then secondly we have to consider for the these diagrams for the Chrysomenidaceae, right? So Chromulina diagram is like this one, or the photographs are there. So you go through it properly. It is given by the Hitachi from the Japan, right? So it is a golden algae. You see, this is the magnified portion of this golden algae. Then the chrysophytes, mainly these golden algae, as you are observing. So these are common microscopic chromista that is found in fresh water. Some species are colorless, but the vast majority are the photosynthetic. Particularly, they are source of the food for the acting as a source of food for the zooplankton. And as therefore number of biologists they have considered as become the facultative heterotrophic for the system or the ecosystem, and they get also grow in the adequate light or the absence or in the some presence of the light. The some dissolved food material generally also requires to them. So therefore the it is mainly used as a predator, right? So there are more of thousands of species of golden algae, and they shows the generally found in the form of swimming as a unicellular or filamentous or colonial forms are there. The Dinobryon is the one freshwater genus, and it shows the face-shaped structure. There are the chitin fibres are there. The colonies grow as branched or the unbranched. Then Cyanura genus, it is colonial genus. In the yesterday lecture we have seen about the diagrams for Cyanura. Then secondly, some according to some researchers, the silica scales they are also present in the Cyanura. In the oldest condition, calcareous and siliceous deposits, that is from the Cretaceous age period. So Miocene period is there. So the fossils are recorded from this period also. As there are some diatoms and the Kukoki lithophroids are there. Litho means that is with reference to the rocks are there. So paleoecological studies also gives about this genus. Then as the Chrysophyta is a heterogeneous group, so probably paraphyletic group is there. So therefore it shows the recognition with that of the Rhapidophyceae, Eumastagophyceae, and Xanthophyceae and Silicoflagellata. So all these are showing the same in characteristics. As mainly Chrysophyceae usually called as the chrysophytes, golden brown algae or the golden algae, and so these are the large groups. Uh, Primnaceum parvum, which causes the fish kill, and so it is a single species of the golden algae. Then although the Chrysophyceae members there are some are there, so according to Smith, they are classified as the order Chrysomonadales into three suborders as per genera. Genera are the Melomonas, Cyanura and Dinobrion. You remember minimum names of the genera. Then Chrysoamoeba, Hydrorus is there, and Pheomethyon or Epicrysis is there, right? Then it is important part in the evolution. The chrysophytes contain the pigment fucoxanthin. Because of this, they were once considered to be a specialized form of cyanobacteria. Right. Then, because of many these organisms have the silica capsules, so they have relatively complete fossil record. And according to modern biologist, to confirm this, in fact, not derived from the cyanobacteria, but rather than an ancestor, did not possess the capability of the photosynthesis. Then there are the most of the biologists believe that the chrysophytes obtain the ability of photosynthesis from the endosymbiotic relationship. Here you see the Bacillariophyta diatoms, so this shows the mostly unicellular aquatic forms. Diatoms are the major group of microalgae. About 5,600 species are there. Mainly diatoms occur in the fresh water, salty water. Some species are attached to the substrates, and pelagically, that is open sea, in open water. Some live as a surface film at the water sediment or attached to the benthic. Then secondly, general characteristic, the cellulose or the cell wall, it is made up of the cellulose in Xanthophyceae. General characteristics, as there is having the cell surrounded by a rigid two parts box like cell wall composed of cilia called as the frustule, and as it is also used with ornamental patterns. Numerous tiny pores are there composed of silica, silicon dioxide, SiO2. All these are reproducing asexually. Then in Bacillophyceae, these are some photographs. So Bacillophyceae diatoms are there. Characteristics according to morphologists. You see the different shape and the sizes. Yesterday lecture we have seen about all this. Then as it is a large group which consisting of 200 genera and 10,000 species are there. 92 genera, about the 569 species are reported from the India, commonly known as diatoms. The diatoms most beautiful microscopic algae due to their structure and the sculpture of their walls are there. Nextly, they occur in various habitats as freshwater, saline water, terrestrial condition, or in the soil also. Sometimes they also occur as epiphytes along with algae on the leaf of the forest trees, and mostly in the tropical rain forest. Depending on the mode of nutrition, they may be photosynthetic autotrophs or the photosynthetic symbionts or the heterotrophs. There are commonly unicellular free living, but they are commonly unicellular free living, but some members from the colonies of the various shapes like filaments mucilaginous colonies. The microscopic cells are different shapes. They may be oval, spherical, triangular. So here just you have to remember the characteristics for this diatom. The cell wall is composed of pectic substances. Yeah, they are impregnated with the high amount of siliceous substance. The wall may be secondary structure like spines and the bristles. The vegetative cells are diploid, whereas the cells generally may be discoid or two large plate-like chromatophores are there. They also show the stellate, that is star-shaped chromatophore. The different pigments like chlorophyll A, chlorophyll C, along with xanthophylls like picoxanthin, diatoxanthin, and the dinoxanthin. Then reserve food is oil, polytin, and the chrysolaminarin. Some vegetative cells shows gliding movement and motile structure. It is shown in the anthrozoid has a single pneumatic flagellum is there. So these are some characteristics for this one. The vegetative multiplication takes place by the cell division, and some cells reduced in size due to which they produce characteristic spores, spores, and develop to regain the normal size. In sexual reproduction takes place by the isogamy or oogamy, and therefore the Frisch 1935 followed the classification of the Husted of the 1930. So Frisch given as the Bacillophyceae centrales. Then there are the different subfamilies, orders are there. Discoidae, solenoidae, as per genera. Genera are the Melocera, Corythron, Bidulphia, Rutilaria. Then order Penels. These are the diatoms. So Cyanandra, Eunocia, Coconeis. Right, genus Navicula, Bacillaria, Suriella. So these are some characters for this one. Then about occurrence, diatoms found in all possible habitats, commonly found in fresh water. Some genera Denticula, Navicula, Meridian, Cimbella. You remember seawater Corythron is the common genera, Fragillaria is the common genera, Pinularia, Navicula, Fistelia. Then terrestrial species Amorpha, Navicula. Then Gomphorina found in the as a diatom in epiphyte. Then Enteromorpha is the name of diatom also. It is also same in other genera. Then Cladophora in the other with other members are there. As on this genera, Enteromorpha is the larger. So on this one they found. Then higher plants. Then Lymphophora, member of diatom, grow endozoically. Endozoically, that is in the stomach of this one. Then plant body of diatom as unicellular moves singly. The cells are different shapes, found oval, elongated, rod shape, right? So all we have to consider this character. So please see these photographs of some. So Rafi is the character. So all these are the single cell. That names are the A diagram Lymphophora, B Bidulphia, C Acranthis, then D Tricheraceum, Casioconidiscus, E, then Eunocia, F, G Coconis placenta, and the H Bacillaria. So you remember do this diagram for our study. Nextly, cell structure. So yesterday we have seen, but here also it shows the hollow and connective and cell shows the covered by a siliceous wall is there. The fistule is the important character. So you see the magnified part. What is the meaning of the whole? Hypotheca, then nucleus, cytoplasm. There are the girdles, hypotheca and raphe are there. Then in the A diagram, you see the cell wall, then wall markings, central nodule, raphe, polar nodule is there at the polar means at the end. So these are the good photographs for this one. Commonly all these characteristics for particularly diatoms, these are to be seen. Then protoplast content, mainly it shows the photosynthetic pigments, chlorophyll A, C, then beta carotene, picoxanthin, and in locomotion, the raphe, diatoms with raphe are motile. Most of the members of order penils contain a raphe and perform the gliding movement. This gliding movement by the circulation of cytoplasm. So here and they also release the mucilage. The rate of movement varies from 2 to 25 micron per second. Right, the locomotion is affected by temperature and the light. So all these about this one. Then in reproduction, diatoms produce vegetatively by binary fission, division, cells 1 to 2, 2 to 4, 4 to 8 like this one. Then sexual reproduction, they show the autogamy, then gametic union is there, develop the auxospores are there, formation in penils. So production of auxospore in one cell, you see how this develop inside to this. Then production of two auxospores by the conjugation cells. So all these are characters for the reproduction. Here one cell, the genus is this Cimbella lanceolata, production of two auxospores. Okay, then production of one auxospores by autogamy, production of auxospore by parthenogenesis. So parthenogenesis means what? As the diploid nucleus undergoes to sequential mitotic division, and this mitotic division with one nucleus as a mitotic degenerate, and therefore thus only one diploid nucleus along with the protoplast remain, and thus it behaves as a auxospores. So perozonium, it's a covering to this which secretes the new wall, and normal size cell is formed. Auxospores by oogamy. Oogamy means different in shape and size. So it behaves as this type. So this is all about the diatoms are there. The auxospore formation by oogamy, it is also one of the important part. Then in fertilization, to form a zygote, the oogonium and it fertilizes with that of the sperm, and later on it becomes a resting spore, and this resting spore reproduce formation of thick walled resting spore, cyst or the statospore. So this is a life cycle. Then in economic importance we are knowing diatomite is there. So uses diatoms. Here the accumulation of thick cell wall is there, and the deposits are called as the diatomaceous earth. Right. So it is very suitable to use for the different industries as a filter we are using. Then it is used as a filter in different industries like sugar, filter microorganisms, oil, and the chemical industry. It is also used in battery boxes. As insulator, it is used heat resistant. As absorbent, used in liquid nitroglycerin. It also other uses like the abrasive, capable to rubbing or the grinding. In petroleum industry, they are found in association with the large oil deposits, and as a food in different seas they are abundant, so used as a food by marine animals. The grasses of the sea, they are commonly called as the grasses of the sea. Those animals may become consumed as food by man and maintain the food chain. And the testing of microscopic lenses, they show the fine marking on the shell. The diatom cell are used to test the microscopic lens. So this is all about our Xanthophyta, diatoms, Chrysophyta, all these one. In next lecture we will see about the Pheophyta. So thanks to all.